Hi, I'm Neha, and today I'm gonna tell you about a very interesting topic that changes biology. Just imagine there's a scientist sitting in his lab named Frederick Griffith. He wants to know that why some bacteria make us sick while others don't. So basically, what he did. So here's Frederick Griffith. He was working on a bacteria named Pneumococcus bacteria, and what he did, he took two strains of the bacteria, S strain bacteria and the R strain bacteria. The S strain bacteria is smooth because it has an outer covering called capsule, and that capsule basically protects the bacteria from the immunity system, and that's why it is harmful. While if we talk about the R strain stand R for rough because this bacteria do not have a smooth outer covering. They have no protection from the immunity system. That's why they are harmless. So what happens when the S strain is injected into the mice? There is a death of mice. While the R strain is injected into the mice survived. Pretty obvious, right? Of course, when the harmful bacteria is injected in the mice will die. And when the harmless bacteria is injected into the mice survive. It is obvious. But when the heat-killed S strain bacteria is injected into the mice survived. Now, it is also normal. When the heat-killed S strain means when the harmful bacteria is heat-killed and injected into the mice, obviously the mice gonna survive. But here's come the most important and most shocking thing. What he did, he take the heat-killed S strain plus R strain and inject it into the mice. Now, what do you think? What do you think? Mice gonna survive or die? The mice died. Could you just imagine? Like what happened when the heat-killed S strain bacteria and the R strain harmless bacteria were injected? The mice died. Wait, how is it possible? Like when we take a harmless bacteria and a harmful bacteria killed, how it can die the mice? How the mice die? Wait, is this something transformed from the heat-killed bacteria to the R strain? But what was it? Well, that's what Frederick Griffith noted. That there's something transformed from the S strain bacteria to the R strain bacteria. And that is called transforming principle. And yeah, we completed a transforming principle. But just imagine what it can be. For a second, take a pause and think about it. You know what? Later, scientists discovered that it is found out to be a DNA. It's DNA. Just imagine how difficult or how shocking this experiment will be for that time. Just how shocking it is for the scientist. And today, we all know that this DNA is a genetic material. So this experiment basically leads to a very shocking experiment that tells that DNA is the genetic material. This simple experiment gives one of the biggest clues that DNA carries genetic information. And this is all. Thank you. Have a nice day.